Okay, guys. Let's get started. So there's a little bit of a panic going on among some students who want to do well, and I promise you, if you're this attentive at this point, you will do well, about some of the quiz questions. And I want you guys to bear with me as I'm changing textbooks from last year, and we're working through redoing some of the quiz questions and altering them and adding new ones and taking away old ones. So bear with me. This is why we keep the 10 best, okay? We will drop, and if there's one where everyone gets something wrong because of something that's unfair, I'm flexible. Don't panic yet. Panic after your first exam when you fail, which you won't do because you're all great students. Okay.
That was so much fun.
Don't worry. I know everyone likes to panic. That's the job of the student. Don't worry. Okay.
So should I not come see
You can come see me if you want. Okay. So quickly before I get started, how is the volume on the recording since we moved to this classroom? Because I'm moving around more.
It's in and out.
Okay. So I'm going to try to figure that out. I'll try to stay a little bit closer to my
Maybe
computer.
if you flip the computer towards.
Ooh, flip the computer towards. We'll see how that works. Okay. But I might need to get a mic. Yes.
Well, we're supposed to get a response back for that extra.
Great. So we're going to talk about extra credit in just a minute. Okay. So, quick business before we move forward. Think about the oral exam option. As you guys know, everyone has an option to do one exam orally. And now is the time for you to make that decision because next week we have an exam and you need to email me so we can schedule it. My guess is not many people will do it the first time, but if you want to try it, then let's do it. It'll be the same exact day of that you would normally take the exam, except for it'll be after 2:30 p.m. And basically you'll come to my office and I'll question you until you cry. No, I'm just kidding. No crying. I'll question you until you can't answer anymore. And based on the level of your answers and how many of the answers you could give me, you will get a grade. So please email me if you're interested. The other thing is we're about a week before the first exam, so I will be accepting multiple choice questions. Anyone who wants to try it. The rules are they must require analytical thinking. No recall questions will be accepted. No all of the above answers. No none of the above answers. All possible answers must be viable. No throwaway answers. And if I use your question, you will get between 1 and 2 points extra credit on your exam plus the value of that question because you should know it if you wrote it. 1 point is if I have to fix it, and 2 points is if it's perfect. And this is not as easy as it sounds. Usually only about 1 or 2 questions get accepted because it takes a lot of work to write a good multiple choice question. So you'll see how that goes. Okay, any questions about the business? Okay, so where were we? We have been talking about chromatin structure and function. So that's the openness or closeness of the DNA and how it relates to different cellular processes, such as gene expression. There are other things that are happening that depend on regulated opening or closing of DNA, but we're mostly using the example of gene expression. We spoke about the concept of post-translational modifications and their role in regulating protein-protein interactions or protein-DNA interactions, and the subsequent effect on cellular functions. We spoke specifically about acetylation and how it's associated with activation of gene expression, and methylation and how it's associated with silencing. And as you'll see as we talk about the extra credit, it's not only associated with silencing, but that's what people think of when you think of methylation. We spoke about protein domains that bind to post-translationally modified proteins, and this is a level of regulation, right? So if you get modification of a protein, then suddenly another protein will be able to interact or not interact, and that will affect cellular functions. We spoke about chromodomains, which bind to methylated lysine, bromodomains that bind to acetylated lysines, and SH2 domains, which bind to phosphotyrosine. And then we spoke about the enzymes that regulate chromatin structure and function. We talked about HACs and HDACs that are involved in acetylation and deacetylation, HMTs and demethylases, methylation and demethylation, and for phosphorylation. What does phosphorylation? I figure I didn't need to tell you. What enzyme phosphorylates? Someone raise your hand because I hear a lot of right answers. Yes?
Kinase.
Kinases. And what dephosphorylates?
Phosphatase.
Right, phosphatases, right. So I do feel like I needed to put that up because you hear about phosphorylation so much as you make your way through this program, and we're not going to focus too much on it, but it is a very important post-translational modification. Okay, then we talked about the concept of the histone code and how these multiple modifications on histone tail mean something. You're going to have lots of methylation at different lysines or acetylation at different lysines, and it creates a code that is read by a complex of proteins and recruits other proteins to do things. And we'll talk a little bit more about this. So this is where we are. So as we start to prepare for next week's exam, and you say, oh my God, what's on the exam? Look at these review slides that I started each lecture out with, and you'll get to see what the main ideas, the main topics you should be focusing on in your studying. Okay, any questions about this stuff so far? Yes?
For the histone code, is that just all of the acetylation, all of them combined together working, or is it just specifically?
So the histone code is created. So the question is, what is the histone code specifically? Is it multiple modifications or just specific modifications, right? So it's multiple modifications happening at the same time on histone tail, and that changes the shape of the protein, which results in recruitment of different proteins to do different things. Very general way of thinking about it. And in recitation this week, we're going to read one paper where someone did a high throughput analysis of the whole genome to look at all the different histone modifications under different conditions, and that's how they started to think about the histone code, because you saw frequently when you have high gene expression, you have these modifications happening. And so we'll read a little bit about that in recitation. Okay, so last week, Stump the Teacher extra credit. So someone stumped me last week. I had my theories about what the answer was, but so we were talking about lysine acetylation and methylation, and I mentioned that you can have monomethyl, dimethyl, or trimethyl lysine. And someone said, well, what's the purpose of these multiple methylations and lysines? Does it have anything to do with the intensity of silencing or tight packing of heterochromatin? What's the purpose? Why are there these different modifications, amounts of methylation? Okay, so a handful of people sent me some nice responses, and I'm going to go over what I found in my research, and we can discuss a little. And pretty much, if you sent me anything that kind of goes with what I found, then you'll get a point. And so the way you can keep track of your extra credit points you get when you stump me is on Blackboard. I'm going to have a column that's cumulative extra credit, and you'll get to see them come up there. It's not up yet, so don't panic because you haven't seen a point there. It's going to go up today. Okay, so what did I find? So I found a few things. I found 2 papers which I posted on Blackboard. They're really nice reviews, and they're more structural biology papers, but this is really about how different modifications affect the structure of proteins and how that affects protein-protein interactions. What I found was that mono, di, and trimethylation increase the diversity of proteins that can be recruited to nucleosomes. Makes sense, right? Each time you add a different methyl group, you're going to change the shape of a protein such that it's going to bind more tightly or not at all or whatever to a different subset of proteins, and that's going to recruit different proteins to the histone to do different things. So it just increases this diversity, and it's a way of regulating chromatin structure. So in this one paper that I posted, They said functionally, histone lysine methylation has been correlated with both gene activation and silencing. So I keep saying it's associated with silencing. Really, it's activation and silencing. It's very confusing, so we won't worry too much about in which case what happened. And methylation states of distinct lysines along the H3 N-terminal tail provide a frequently cited example of this diversity. So there could be lots of different methylation and methylation at different levels, and it does different things. Within this defined region, H3 lysine 4 methyl. So the way they annotate it, so that's the histone, that's the lysine number, and that's how many methyl groups. So histone H3 acetylated at lysine 4 with 3, and not acetylated, methylated at lysine 4 with 3 methyl groups. Is highly correlated with active transcription start sites. If you're methylated with 3 methyl groups at lysine 9, it's linked to constitutive heterochromatin. Very different, right? And if it's methylated with 3 methyl groups at lysine 27, it's linked to silent genes and euchromatin or facultative heterochromatin. So basically, I put this up here because it was basically saying that there's a lot of different functions of methylation, and that if you're mono, di, or tri-methylated, it just increases the diversity of the types of proteins that can be recruited in order to activate transcription or move nucleosomes or whatever. So I have a few figures I wanted to show you. So the way to think about this is that, remember we talked about chromodomains and how they recognize methylated lysine, right? Everyone remember that last week? And I had Legos. We talked about Legos. Who wished that they could play with the Legos? Yeah, okay, good. I like honesty in my students. Okay, so in this paper, they basically show that there are different domains. So I talk about chromodomains. It's not only chromodomains. There's a whole family of domains, multiple domains that bind to methylated lysines. And some bind to dimethyl lysine, some bind to trimethyl lysine, some bind to monomethyl lysine. So what does that mean to you? That means that there's all these different proteins that are specifically going to be recruited to one form or the other. And so this is just showing, so there's your methylated lysine, and then there's this this structure that likes to interact with it, and it's showing here there's your trimethylated lysine and how it's interacting with a double chromodomain. Here's a chromodomain, and that methylated lysine binds right there. Whatever, you don't need, you need to memorize this for tomorrow, because you have to draw these all out. Okay. No, I just want you guys to understand that that proteins have different shapes, and they're going to interact with other proteins of different shapes, and when you get modification, that's going to regulate that interaction. So this was another nice figure that they had that kind of showed that how there are these different levels of methylation, and it doesn't change the charge of the lysine, right? So they all remain positively charged, not like acetylation, which actually gets rid of charge. But it does change the shape, right? You can imagine, for those of you who love chemistry and biochemistry, or just like to think about molecules, if you think about lysine, right, this is what lysine looks like, and here's the end of it where you get methylation. Lysine normally looks like that, where each of those is a hydrogen, and as you add a methyl group, it changes the shape. See, the green is the methyl group. Here's one methyl group, 2 methyl groups, 3 methyl groups. It's changing the shape, not the charge really, but the shape of the lysine, and that's going to affect the binding pocket. And so this is showing all these different domains and proteins that prefer different post-translational modifications. So you can see there's a huge amount of diversity. And this picture I thought was really nice. So this was from a paper where they were looking at lysines that are methylated at different levels. So there's unmodified 1-methyl, 2-methyl, and 3-methyl groups, and they're looking at the binding of the different proteins, okay? And so the way you can look at this, so this is a protein right here, And here's the binding pocket for the methylated lysine. And so don't forget this methylated lysine is attached to another protein. For simplicity, we're just showing the methylated lysine, but that protein-protein interaction is actually, it's two proteins, it's not just a modified amino acid, right? So you can imagine this lysine prefers, this binding pocket prefers to bind to dimethylated lysine. And so here they actually changed them and looked at how tightly the binding is. And so when you have unmethylated lysine, there's a very low percentage is bound. And then as you change your lysines, you get to an ideal. So with, here with mono and dimethylated lysine, the most protein binds. And so it's just a way, the point I'm trying to drive home is it's just a way to regulate protein-protein interaction, to recruit proteins to chromatin, to do whatever needs to be done, be it modified nucleosomes, yes.
So with having the trimethyl, would that kind of hide the positive charge of the protein-protein interaction? Would be harder for the.
So would the trimethyl group hide the positive charge of the protein-protein interaction? Well, so what it said in this paper is that methylation doesn't really alter the charge, it remains positive. But if you think about the size of a methyl group compared to a hydrogen, it's changing the shape, right, on that surface of the molecule. So it's gonna really either it's gonna fit into the binding pocket or it's not. Okay, thanks. Any questions about this? I know I went into a lot of detail, but I felt that the more I thought about the topic that came up in class on Friday, the more I don't know, detail helps me understand. Go ahead. So you're saying, wait, so repeat that? It's associated with silencing and activation. So, so that's kind of a way to understand it. Yeah, you can have monodi or trimethylation. They take different paths to end in stuff, but not always silencing. Silencing, activation, any of that list of stuff that I had on the last slide. But it's different paths. I like the way you said that because you're recruiting different proteins that are going to take the function in one direction or the other. Yes?
So if it's trimethylated, is that able to come within a acetylate bit, or is it blocked from acetylate?
If it's trimethylated, is it blocked from acetylation? Is that what you're saying? So it's the same lysine that's going to be acetylated. So in order for acetylation to happen, something needs to recruit a demethylase to remove those methyl groups to acetylate. Yep.
All right. The red and the green line are mono and dimethylated.
Yep.
So doesn't the dimethylation also sort of upregulate the whatever? Because I can't think about how to say it, but a demethylase has to take off two methyl groups in order for it to stop the binding affinity. So it's also upregulating that by having two on there.
Okay, so you're saying so you're thinking about the activity of a demethylase in removing methyl groups and how if you have two methyl groups on there, it's going to maybe bind more strongly because it takes more to remove them?
Yeah.
Is that what you're thinking?
Overall, there's going to be more.
Yeah, I don't know. That's interesting. It's an interesting thought.
Okay.
Yeah, go ahead.
My question is, if you're using HMTs to methylate, wouldn't it depend on the enzyme that you're using, whether or not it will actually bind to a protein? Because the enzyme will change the overall charge of the molecule, but if the enzyme function is to only work within a specific charge, would it not matter if it's mono or trimethylated because of the enzyme's function?
Okay, so you're saying the enzyme that methylates, so are you saying is it based on the enzyme's function? And so does it not even matter because the enzyme's going to do mono, di, or tri because it's the job of the enzyme. So it's specific. So there are many methylases. So probably what happens is in certain conditions, a methylase that likes to dimethylate is going to bind. a methylate acetylase, a trimethylate is going to bind. And so that's the level of regulation. You can take it back a step. It's not only that you'll have multiple methyl groups added, it's even before that, which methylase is being recruited in order to methylate either mono, di, or tri, and then that's going to influence the interaction. Does that make sense? So you're right, but it's not like it's just by chance. It's regulated even at that level. Okay, before we move on, any last questions? Okay, so thank you guys for stumping me and making me read all about this. I appreciate that. Okay, so for those of you who are like, whoa, what is she talking about? I'm guessing someone out there is thinking that. Let's take it back to the big picture, right? Because we're getting bogged down with the detail of how many methyl groups are being added to a lysine, right? So let's take it back and let's just think, how is this a way to regulate cellular processes? Okay, we're talking about modifications, different modifications, right? And the whole point of the whole methylation and acetylation and phosphorylation, all that stuff, is about recruiting different proteins that are going to do things, right? So how is this diversity in the number of methyl groups that can be added to a lysine, or even an acetyl group that can be added to a lysine, how is this a way to regulate cellular processes? Big picture. As big as you can get when you're stuck in the chromatin. Yes?
You need a lot of proteins to do a lot of different things, and if you only have so many ways to edit modification with the methylation, then you're going to need to have different ways to methylate to produce all these different proteins that you need. So it just it creates a lot of variation. So with that variation comes many possibilities of change.
Okay, so I'm hearing that you need a lot of different proteins to do a lot of different things, and with all this variation in post-translational modification, that allows for different proteins to be recruited under different conditions. Is that pretty much what you're saying? Anyone want to add to that? Yes?
Well, since there's an ideal type of modification for interaction with each protein, that means that it can kind of sort of interact with other kinds of modifications that aren't ideal. So I guess, like, instead of it just being an on-off switch, it's more of like a dimmer switch.
Oh, I like that. Right, so there's an ideal modification that's going to allow different interactions to happen for each group of proteins that needs to interact. Don't forget, this is all about interaction with histones, right? And so rather it being just on or off, like methylated, no interaction, acetylated interaction, it's kind of like a dimmer switch where you can be more fine-tuning what types of proteins are going to be recruited. Yes?
I'm just kind of confused. I thought that methylation and acetylation were more of the chromatin's open or closed, and then it's also associated with proteins. So it's like, what's going on there? Like, it's open and then it's recruiting proteins to have something to transcribe, and that's adding to it, like at the same time.
Right, so your question is, you thought of methylation and acetylation as whether or not that chromatin's open or closed, right? Right, so this is getting to that point, right? So methylation will happen, and so to go back to the big picture, absolutely, that will recruit proteins. One might be, and what we're going to talk about today, one might be a nucleosome remodeling complex, which is going to move your nucleosomes around and then expose transcription factor binding sites and stuff like that, so gene expression can happen. So what we're talking about is recruiting the proteins that are going to help to change the architecture of the chromatin so gene expression can happen. Make sense?
Proteins are just different ways of opening.
That's as far as I understand, the only way to open and close chromatin is through recruitment of proteins that actively move things around. There is the very basic acetylation of a histone core. It's going to change the charge and loosen up the DNA through that abolishing that positive-negative interaction. So that's definitely part of it, but it's more than that, because then when it's loose, you still need to slide your histones around. Okay? Any other questions I saw? Yeah.
There's so much you said about acetylation. Do they know the fixed number of acetylate lysines that have that purpose to have forever or to use it now? Or is it very between nucleosomes?
Okay, so is there a certain number of acetylated lysines that need to occur in order to open up the DNA for something to happen? I have no idea. You guys are trying to stump me now. Actually, I do know. So in the paper we're going to read this week in recitation, that's what they're looking at. They're looking at the types of modifications that are happening on histone tails under different conditions, such as active gene expression. So when we read that paper, I think we should pay attention to that, see how much acetylation is happening. Yeah. Any other questions? Okay, that was a rousing discussion. Okay, good times. Okay, so we'll move on. Oh, right. So the other model, and this paper came up, and I had to say something about it because it's written by someone who's people who I kind of have known in the past and I really respect their way of thinking, though this appears to be like a less complex model in a way. It's that modifications directly affect chromatin structure. It's the regulated nucleosome mobility model, which is about how you can have a modification and that will result in either low mobility or high mobility nucleosomes. High mobility means they slide around more, and low mobility means not so much. So you can imagine high mobility would be associated with activation, and low mobility would be associated with repression, because in order to have activation, you need to be able to slide your nucleosomes around your center. So I'm just going to give a moment there, but it's kind of similar in a way. It's just another way to think about it. Okay. So if heterochromatin is DNA that is tightly packed into chromatin and is not available for transcription, what needs to happen for transcription to occur? So tightly packed, closed DNA. What needs to happen so transcription can occur? Yep.
Just somehow the proteins are acetylated.
Okay, good. Bigger picture. So something about acetylation, but bigger picture. What needs to happen to the chromatin? It needs to open up, right? So, yep.
Oh, that's okay.
Okay, so the chromatin needs to open up. So you're talking about the mechanism by which it opens. Right. So what kind of proteins might be required to open up the chromatin so that it's available for transcription factors and RNA polymerase and all those important things to come in? Yes. Hats. Sorry. We need to wear hats. Okay. So where would you buy that hat? Nordstrom? Specialty store? Online? Okay. Right, so hats. So one thing you would need is a histone acetyltransferase, because that would do what? Thank you. Acetylate the histones, which would do what to the tightness of the DNA? It would start to loosen it up because the DNA won't bind very strongly to the histones. Right. So what else might acetylation do? Let's say you have acetylation on a histone tail. What kind of protein might that recruit? Yes? A protein containing a bromodomain, absolutely. And what kind of protein containing a bromodomain might bind to acetylated histone to activate transcription? Don't worry, I'll tell you in a minute. Chromatin remodeling complexes, right. So sometimes I'll say nucleosome remodeling, sometimes I'll say chromatin remodeling, because depending on the textbook you read, those are interchangeable. But the purpose is to slide those nucleosomes around to expose promoter regions so that DNA binding proteins can come in. Okay, so that brings me to this question, which I'm putting up because there's some confusion about it. Nucleosome remodeling complexes participate in movement of histones to increase accessibility of DNA prior to transcription. How do nucleosome remodeling complexes know which nucleosomes to move? Forget about the answers for a minute and just think about the question, right? So you need to move nucleosomes around so you can get to your promoter. how do the nucleosome remodeling complexes know which nucleosomes to bind to and move? Yes?
Maybe ones that have certain modifications on them.
Ones that have certain modifications on them. Absolutely. I saw another. Right. Okay. So what kind of modifications? Acetylation or methylation, because they can move something in the way or out of the way, right? You can think about it that way, right? You can move it out of the way of a transcriptional start site, right? Okay, so as we're talking about all these modifications and we start to get kind of bogged down with the details of how many methyl groups are added, remember, a lot of it has to do with changing the shape of your histone to recruit different proteins that will alter the structure of the chromatin so it's available for binding to transcription factors, pretty much, or other things. I'm using gene expression as an example a lot. Okay, so the answer to this was B and C. And I apologize for that, because I think it caused a little confusion. Obviously it was not A. They remove all histones from the DNA allowing access to any gene of interest. That's a crazy answer, right? Do you need to unpack all of your DNA just to transcribe one gene? No. Okay, so that was the easy no answer. Nucleosome remodeling complexes often contain bromodomains or chromodomains and are targeted to acetylated or methylated histones. Absolutely, right? But then there's another part that actually is not in this textbook. And that's where I apologize to you guys, because in changing textbooks, I lost some information. And now I feel like I need to use both textbooks, but we're not going to because I hate that textbook. Nucleosome remodeling complexes are recruited by histone-interacting proteins via acetylation-dependent or independent mechanism. And what this is saying is that you can have modification on histones that will recruit nucleosome remodeling complexes, but you could also have a protein that will bind to the DNA in a specific spot that will recruit nucleosome remodeling complexes. So we'll learn this, and it'll be in the lecture, but it's not in Albert's. I just searched for it, and I was very disappointed. I guess nothing's perfect. Okay, any questions about this? Yes?
In the model of sliding histones to open up DNA, can't you technically only slide so far to open up only a specific amount of DNA?
Okay, so in the model of sliding histones to open up the DNA, can you only slide so far? So you're thinking of it as literally beads on a string where you're going to eventually butt up against another bead, right? Well, so that's where there are two models, one sliding and one's removal. And I'm not sure which has the most evidence, but that would tend to favor the removal of histones, right? Because then you don't have that problem. Don't forget, it's not one nucleosome. Often, many nucleosome remodeling complexes will be recruited to many histones, and so there's a lot of movement. Okay. Any other questions about this? Okay. So, chromatin remodeling alters the location of nucleosomes on the DNA. So, here's your beads on a string for simplicity, your histones with your DNA wrapped around it, and this pink part is going to be your DNA binding site where your transcription factor needs to bind in order to activate gene expression. And as you can see, is it going to be easy for a transcription factor to interact with the promoter here? Is there any accessibility? No, it's tightly wrapped around a histone. There's no way for a transcription factor to get in there and bind and activate gene expression. Well, maybe there's a way, but not that I know. So something needs to happen to move these nucleosomes out of the way, either by sliding them or taking them off and then putting them back on elsewhere. And that allows exposure of your DNA binding site, which sometimes I'll refer to as a promoter because that's often where transcription factors will bind, or an enhancer region. And that allows the DNA binding protein to come in and bind. Okay, so that's the big picture. actually the bigger picture would be for you guys to really always keep in mind this all has to do with whether or not gene expression can be on or off. Okay? So you have to think about nucleosomes as being dynamic, right? They need to move to expose transcription factor binding sites, promoter and enhancer regions. They're extremely dynamic. There's this figure in the book that I liked. So a wrapped nucleosome exists for 250 milliseconds. That's really short, right? And then it will unwrap for 10 to 50 milliseconds, and then it will become rewrapped again. And during that time, that exposes the promoter and allows some kind of transcription factor to come in and bind. This constant movement, yes?
Would this shift with the acetylation of the histone, like more time is spent unwrapped?
So if the histone's acetylated, would there be more time spent unwrapped? So don't forget, these modifications are all reversible. So acetylation probably needs to happen because the part that you're missing in this beautiful picture is the fact that you do get acetylation, which results in loosening, which then results in other, you'll also get other acetylation, which will result in recruitment of bromodomain containing proteins that are going to then move the histones out of the way. So it's part of a larger function. And then you're going to have deacetylation at some point because you're going to close this back up. Okay, any other questions about this concept? So don't ever think of anything as just like acetylation happens and that's it. It's just this dynamic, nucleosomes are very dynamic. They're constantly moving around because your genome is constantly, different parts of your genome constantly need to be accessed to express different genes, right? Yes.
So in some cases, the binding protein will fit its way in between the histone and the DNA, and sometimes it doesn't. Is that what it's showing? When I feel like there's two different paths.
Oh no, so what happens is you have your tightly wrapped histone, and then it becomes unwrapped, and that allows your DNA binding protein to interact with and bind. And then when that's gone, then it will become rewrapped again. Yep. And probably what happens is there's deacetylation and recruitment of other proteins. So this is a very simplistic picture. Okay. Any questions about this? Are we having fun today? Did anyone have a good weekend? Everyone says yes. Did anyone have a really bad weekend? Yeah, I'm sorry about that. We'll try to make for a better weekend next weekend. I always say to myself when it's a really bad day, well, this day's going to end eventually. Tomorrow will be better, right? And then just go to sleep. Okay. I tell that to my kids. I'm like, well, time marches on. Okay. So nucleosomes are moved around on the DNA by ATP-dependent nucleosome remodeling complexes. So ATP is required for the nucleosomes to be removed. Okay, so this is another figure showing that ATP-dependent chromatin remodeling complex comes and it binds to a nucleosome that's probably been modified in some way or has some protein that's recruiting it. It's not random, it's highly regulated. And this results in the sliding or removal, depending on what model you choose to follow, of nucleosomes in order to expose different regions of the DNA. Okay, so these ATP-dependent nucleosome remodeling complexes are multi-subunit enzymes, and they move the nucleosomes along the DNA or remove them from the DNA to increase accessibility for transcription. So here's 4 different nucleosome remodeling complexes, and what's highlighted here are conserved domains. Okay? Again, I'm talking about domains, conserved domains. When you think about domains, you think about function, right? Because these are functional units of a protein. And so if you look at the domain organization of these different proteins, you kind of learn a lot. So you've got the red is these DEX or helici domains, and these are ATPase domains. So if you're studying a protein and you blast it and you find a certain domain, usually that domain has some kind of function, and you can learn a lot about your protein by looking at the domains, right? Some have bromodomains, some have chromodomains. Why would that be? Why would nucleosome remodeling complexes have chromodomains or bromodomains? I know it seems like a trick question, but it's just so easy because I've been harping it for a week and a half. Because they're going to nucleosome remodeling complexes are going to be recruited to histones that are modified in a certain way, right? Then there's these slide, and they bind to histone tails. Makes sense because you're going to interact with a histone, right? So if you have a chromatin remodeling complex which has a bromodomain, is it likely to be associated with activating or silencing? A bromodomain. Activating, yeah, right. There's going to be, because it's going to bind to an acetylated lysine on a histone, and then it's going to probably shift the nucleosome out of the way so to open up and expose some kind of DNA that's needed. Okay, so how can we predict the function of proteins by looking at the conserved domains they contain? Yes?
Based on the domains, you can tell what has happened to either to the histone or its DNA. So based on the marker that's there, you can get a specific domain, and based on that domain, just
Okay, so I'm really glad you gave me the explanation because it makes me think I need to explain this in a different way. So domains are going to do things, right? They're functional units of a protein. So a domain that has ATPase activity is going to do what? What's huh? Right, hydrolyze ATP. Use ATP. Remember I said these are ATP-dependent nucleosome remodeling complexes. A domain, a chromodomain is going to bind to a methylated lysine on a nucleosome, right? And so knowing something about the functions of these conserved domains, and they're conserved in that they're present in many different proteins. You can see they're present in these four different proteins. So you can say something about the job of the protein by looking at its qualifications. It's kind of like the resume of the protein, right? You can look at it and be like, oh, well, you've hydrolyzed ATP before, and you've bound to acetylated lysine before. Oh, and I see you've bound to a histone tail. Oh, you will probably make a great nucleosome remodeling complex, right? Do you think that nucleosome remodeling enzymes go to job interviews? Okay. So I want you guys to really understand this concept of domains. Please see me if you're so confused by it. Okay. How are we doing on time? We still have time. Okay. So nucleosome remodeling complexes, along with histone chaperones, and chaperones are exactly what they sound like, right? When you go to a dance and you have to have a chaperone, usually means there's an adult with you, right? So a chaperone is a protein that likes to accompany other proteins to do their job. They alter the position of nucleosomes on the DNA by either sliding histones along the DNA or by removing histones and reassembling them somewhere else on the DNA. And they're also used to remove histone dimers and replace them with nucleosome, histone variants as needed. So here they're showing two different things. Here's your nucleosome remodeling complex, and there's a histone chaperone, and it helps to remove part of the histone, so in this case H2A and H2B dimer, and exchange them with histone variants. Remember we talked about histone variants briefly, and we're going to talk about them again in a minute. Or you can have a histone chaperone come along and help to completely remove the histone. Yes?
Kind of like the question you answered in the class. Does the number of domains you have affect anything?
Does the number of domains that you have affect anything? Well, since domains have different functions, if you're a huge protein with many different domains, you probably have many different functions. So for example, some viral proteins, which are my favorite kinds of proteins, have tons of domains and do things everywhere, everything from bind to DNA to ubiquitinate other proteins to activate gene expression because it depends on these different little modules that are in the protein. That's pretty much the only way having a lot of domains. So the number of domains is proportional to the number of jobs a protein can do. So if you see a protein, like ubiquitin is a really small protein, it's like 76 amino acids. It has a ubiquitin fold, it has a ubiquitin domain. That's its job, to act as ubiquitin and ubiquitinate things. But like some other proteins will have DNA binding domains, will have transcriptional activation domains, and that might be 2 jobs. And these will have multiple jobs, but maybe all those jobs come together to do one function. So if you bind to a histone and you bind to an acetylated lysine and you hydrolyze ATP, those are all things that need to happen to have an active nucleosome remodeling complex. Does that make sense? Okay. Questions? Comments? Concerns? I don't want to hear it. No concerns allowed. Okay. So just to remind everyone about histone variants, these are these histone, they're histone variants. They're histone subunits that will replace some of the normal histones in the nucleosome under certain conditions, right? So there's this histone H3.3, which looks just like H3, but it's involved in transcriptional activation. There's SEMP-A, which is it's involved in centromere function and kinetochore assembly. There's H2AX involved in DNA repair and recombination. So you can imagine, like, remember, your genome is super dynamic, and there's this constant, all this constant change in architecture. There's also change in your histones that's going to help to recruit different proteins to do different things under different conditions. I'm purposely vague because there's so many things that can happen, right? So the example that we're going to do right now is H2AX. And so we're going to end class with a short practice problem. We're going to make you guys think really hard. Okay, so you are working at a pharmaceutical company trying to identify new chemotherapy for cancer treatment. So you've all graduated from Towson, and then you went and got your PhDs. And so now you're working at a drug company, and you're trying to find new cancer drugs. You know that the histone variant H2AX is associated with DNA damage and repair, and CHEK2 is also associated with this. It's recruited to these DNA damage foci during DNA damage response. So H2AX, histone variant, important for DNA damage response. And same with CHEK2. So a good cancer drug, by definition, in this class of cancer drugs, will cause DNA damage because DNA damage causes apoptosis and cell cycle arrest. Do you want to kill cancer cells? Yes, right. So a lot of the time you induce damage in DNA to activate the DNA damage response, and that stops the cell cycle. All these red flags of the alarms go off and you die. Well, the cell dies. The organism survives. So you're screening cells treated with drugs. And so what you're looking at here is cells treated with compound X. And this is one cell, so here's DAPI staining your nucleus. Then this is CHEK2, one of the DNA damage proteins, H2AX, your histone variant, and this is the merge of all three. And when you see it lit up, it means your antibody is binding. It means that protein is there. Okay? So stop, talk to your friends for the next 1 minute, and tell me, is this drug a good candidate? Talk to your friends. Where your hands.
There's specific stage regions. So that one common state for
I think of like big because I just get the glue from the very top. I saw that there and it shows up there. Like 2 very well, I think that it's but I feel like the answer can't be that. I'm trying to figure out like what. Like it's just more work than something
Okay, guys. So I'm sorry to cut this short because I do hear a conversation and it's about science, which makes me happy, rather than about the Ravens. But no one's going to talk about the Ravens anymore because that's over, right? No, people are still talking about the Ravens. Okay, we're happy, we're excited. Okay, so the first thing you're going to tell me is that there's a control missing, which is the untreated cells, so you can see what that looks like. And I'm sorry. But basically, untreated cells will have nothing here and nothing here. Okay? So is this a good candidate? Who says yes? Raise your hand. Okay. Who wants to tell me why they think it's a good candidate? Yes.
The two variants, or variant CHUK2 and the H2A are associated with each other, so they're working together to start the DNA.
Okay, so CHEK2 and H2AX are co-localizing. They're in the same spots, which means that CHEK2 is being recruited to histones that have H2AX, and H2AX is only there when there's DNA damage, because that's what recruits DNA damage proteins to fix the DNA. So this drug has induced some nice DNA damage, because look at all that H2AX, tons of H2AX and tons of CHEK2. So I say we should move forward with this drug. Okay? And then I'll call my lawyer, you call yours, and we'll make some money. Okay, thanks guys.
You don't mean it's causing damage, you mean it's found the damage.
It's causing damage.
You want it to cause damage?
Because you want to kill your cancer cells. I know it required a little more intro than I gave, probably.